Our final chapter for the semester then is taking a look at evolutionary history in the very large sense, looking at some large patterns, how do we trace evolutionary history, and some major events that have happened in the history of life on our planet. So if we think big picture, we know that the Earth is updated at about 4 billion, and that's billion with a B, years old, so an extremely old planet. The conditions on Earth were very extreme compared to today, and certainly the environment and the atmosphere was a very different place than what we have today some 4 billion years later. We know comparatively it was a very hot and hostile environment, that as that new Earth was forming, physicists tell us that there was a tremendous amount of geologic activity, volcanic activity, movement of the Earth's crust, earthquakes, those sorts of things. And we know that our atmosphere was very thin. It didn't contain the amount and the types of gases that we have in our atmosphere today. And certainly one of the main aspects of our atmosphere today is it blocks out a lot of that UV radiation from the sun. And so without that atmosphere, that UV radiation would bombard the Earth unblocked, and again, a very hostile, very hot sort of place. Now the major difference in terms of the gases in the atmosphere had to do with oxygen. And we know that O2, or oxygen gas, is a major component of our atmosphere today, and is certainly very important to organisms like ourselves that are aerobic and that need to have oxygen as one of our nutrients. And we know this because if we look at the fossil record, older rocks, especially rocks after about 2.5 to 3 billion years old, we see very little rusting of the iron in those rocks. And rocks younger than that, we start to see more and more rust associated with those rocks. And we know rust is a result of iron being oxidized or mixing with O2 gas. And so we have a good idea of oxygen levels on our planet. And we know that basically it was lacking at the very beginning, that our atmosphere was made up of the same types of chemicals that we see being spewed from volcanoes that were forming that early atmosphere, things like water vapor and methane gas, carbon dioxide and ammonia, a pretty toxic mixture of different gases. And as that atmosphere formed, the water vapor was cooling, and that cooling water vapor then, as it went higher up off the Earth's surface, would turn into rain, and we start to see Earth's very first primitive bodies of water and primitive oceans starting to form. Now I do want to point out, as we start talking and thinking about life's origins, certainly we have very little actual data looking at life's origins. We do have some data in some experiments, and we're going to talk about some of those, but all of this information we're going to talk about certainly is at that hypothetical level. These are hypotheses. They're supported by a small amount of data, but we certainly don't have a tremendous amount of data looking at these kinds of issues. Now realize, as we talk about our theory of evolution, that theory, we don't use that word often in biology, means an idea or hypothesis that's been tested hundreds or thousands of times and we have a lot of confidence in. The theory of evolution tells us what happens to life once life is here. The theory of evolution really doesn't apply to the origins of life or try to describe how did life originate. And so there are certainly many hypotheses. Maybe life arose from a meteor, a bacteria stuck in the middle of a meteorite that hit our planet. Maybe that was our first life. The hypotheses we're going to look at that has the most support, though, is that life originated on our planet from non-life. And there's a whole series of experiments plus observations in the fossil record that lead us to this basic idea that life evolved from non-life. And the idea here is that, again, from that last slide, physicists can describe, here's what the early Earth was like. Here are some of the chemicals that we see in the atmosphere, the heat and the UV light and the volcanic activity and so on. Now, we do know that life existed by about 3.5 billion years ago, and all of our oldest fossils date around 2.5 to 3.5 billion years old. All those oldest fossils are single-celled prokaryotic organisms, so they're very small cells. They're cells that are very similar looking, at least, to bacteria. And so here's some examples. We can see these little round circles represent our prokaryotic cells. Some of them are free-swimming like we see here in this particular fossil. Others grew in very thick mats called stromatoliths. And so here's a stromatolith that we can see lots of different layers, and each layer contained thousands of these single-celled prokaryotic organisms. So again, we know that life was here by 3.5 billion years ago, and once it was here, evolution has acted on that life. But we're going to look at the hypothesis, how did that life originate? Well, the first step for life is to have macromolecules, to see some of the chemicals that we see in living cells, things like sugars and fats and proteins and nucleic acids and so on. And we know that if we take a mixture of the different methane gases and the carbon dioxides and the water vapor, and we add energy such as a spark to simulate lightning, for example, or to simulate that UV radiation, experiments have shown us that those mixtures of chemicals will react together, start to produce amino acids and nucleotides, the basic building blocks or foundations of life. And Henry Miller in the 1950s was the first to do these kinds of experiments. Many have been done since Henry Miller, kind of expanding on his basic idea. But again, he kind of took these different chemicals, mixed them together in a way that physicists told us these chemicals existed, and sure enough, we see these basic building blocks of life start to originate. Now again, these things wouldn't originate today, or couldn't today, because oxygen in our atmosphere is very detrimental and would break down these chemicals as fast as they formed. But in a non-oxygen environment, these chemicals would form and be relatively stable and exist in the Earth's oceans. Carrying on Miller's experiments, we see that these small organic chemicals like amino acids and nucleotides would then combine to start forming larger polymers, the first very basic proteins and nucleic acids that we might expect to see. And again, several different experiments have shown if we mix these things together under these conditions, we start to see these basic chemicals starting to form. And these chemicals, once they spontaneously form, they would exist and be stable. Sometimes you may have heard the term primordial soup. What that refers to is that idea of these shallow ancient oceans containing lots of these organic chemicals that would represent the building blocks necessary, at least, to make life. The next step for life then would be the formation of what are called protobionts. And protobionts just refer to sort of pre-cells. And we know that cell membranes are made up of phospholipids, and these phospholipids or fats can form in these primordial soup conditions, in these very basic primitive chemical conditions. And we know that these phospholipids on their own, if we toss them even today in a beaker of water, these phospholipids form a small little sphere-like structure that looks very much like a cell and a cell membrane. And here we can see under the microscope these are several different phospholipid spheres that have formed on their own. They're not alive, they're just collections of chemicals, but they do look very cell-like. And so the idea is, as these phospholipid spheres or protobionts were forming, the internal chemical environment would be separated from the external chemical environment. And we know that these phospholipid spheres, again, will change shape by osmosis, by gaining or losing water as water moves in and out of the cells, and that they're selectively permeable. Some chemicals can move in, some chemicals can move out. And so a protobiont specifically is our phospholipid sphere that would contain some of these primitive macromolecules, some of these basic amino acids and proteins and nucleic acids and so on. And we know that that internal environment, then chemical environment of that protobiont would be different than the external environment. And again, while no one experiment has kind of taken us through all these steps, individual experiments have shown us that all of these small steps are certainly possible based on these chemical conditions. Finally, our third major step would be to have self-replication. The thing about life that separates us from non-life is our cells can replicate and copy themselves, and the chemicals, in particular DNA in our cells, can copy itself and pass that on in inheritance to the next generation. And so we think that based on the chemistry that we have RNA beginnings. We know that today RNA is relegated to sort of that protein building role. But if we take a look at RNA as a chemical in cells, we're starting to learn more and more about RNA, and we know it has many functions. Some RNA acts like DNA. It can replicate or copy itself. Other RNAs can act like proteins. They serve as enzymes to help speed up metabolism and cause chemical reactions to occur. And so because RNA has some basic functions similar to both of these chemicals, it's thought that RNA was probably our first genetic material, our first self-replicating material. And experiments again have shown that if we take RNA and we mix just a piece of RNA with several different nucleotides, the A, U, G, and C building blocks of RNA, that these nucleotides will start to attach to the RNA and self-replicate, start making copies of that piece of RNA. And so that first cell, that primitive first form of life, was probably a protobiont, one of these phospholipids with lots of different macromolecules inside, that developed a piece of self-replicating RNA. And then that gradually over time, evolution would act on that first cell to make that cell more efficient at life. And early ideas about the first types of life thought that, well, the first types of life must have been photosynthetic because there was no other life there to eat, and so they must be able to make their own food. Today we know that that's probably not the case. Photosynthesis is a really complicated chemical pathway, and so that probably didn't evolve or show up in cells until much, much later in the history of life. It's thought that those first cells, those self-replicating protobionts, were most likely to be organisms that eat things for food, heterotrophs, and it's thought that they ate lots of that primordial soup, that while all those macromolecules were floating around, different proteins and amino acids and nucleic acid building blocks and fats, there was plenty to eat of these chemicals that were being formed. And so these early cells could have digested these chemicals, used them for energy to replicate their RNA, to build their basic chemicals. And again, our origins of life are really separate from evolution because it's not really contained or described in our theory of evolution, and very much at the hypothetical level. But once we get life, we have a tremendous amount of evidence showing us how evolution has proceeded, what are some of the major stages. And we talked about the fossil record last chapter, so I'm not going to get into depth, but I do want to point out the fossil record really does document the basic history of life. Now again, we know it's incomplete, so we're not going to see every species or every step along the way, but we're certainly going to see the major milestones. What did organisms look like during different periods of time? How much diversity was there? Issues regarding speciation and extinction, again, that we mentioned in Chapter 14, are all major issues. And as we can see on this slide, a tremendous number of fossils, literally millions upon millions, have been collected and studied and described, and many more millions are on the way that people have collected and haven't yet described or studied in detail. So here we can see things like this fossilized fish, one of the first vertebrates. We see a fossilized trilobite there on the left, a very primitive insect or arthropod, a fossilized fern, and then on the right there is a fossilized primate, thought to be perhaps a link that eventually led to the anthropoids and eventually us as humans. Now, one important aspect of the fossil record is to be able to date fossils, to be able to, if we find a particular fossil, have a general idea, when did this organism live, where does it fit into the big picture in terms of that history of life. And to do that, we can date fossils by what's called radiometric dating. And radiometric dating uses radioactive isotopes to be able to date fossils. And we know that radioactive isotopes exist in our environment, whether it's the rocks or the air or even living organisms like ourselves, that we all have small amounts of these radioactive isotopes that tend to be unstable. And so radioactive isotopes are going to break down at a consistent or constant rate back into their stable form. And as they do that, it doesn't matter about the environment. It doesn't matter whether that isotope is in the sun or in the shade or is buried 10 miles deep in the ocean, that these isotopes break down at a consistent and steady rate. And the rate they break down is measured in the half-life. And different isotopes have very different lengths or half-lives in terms of how long does it take this isotope to break down. Some isotopes, like carbon-14, are very unstable, and so they break down relatively quickly. And so carbon-14 has a half-life of about 5,700 years, which means if I have a gram of carbon-14 in 5,700 years, I'd have half a gram of carbon-14, and that other half of the gram, the amount that broke down, would be back into carbon-12, the stable, more common form that we see listed on periodic tables, for example. And so 99+% of our carbon is made up of carbon-12, but because we're living things, about 1% of our carbon that we take in is this carbon-14. Other isotopes are more stable, they break down much more slowly, and so they're going to be used to date things that are much older. So for example, potassium-40 is a stable isotope. It has a half-life of over 1 billion years, and so it breaks down very, very slowly into its stable form, and so we can use that to date things that are much, much older than something like carbon-14, which we would only date something maybe 50 to 60,000 years old or younger. And the idea is that the older something is, the more of the isotope has broken down into stable form. So the amount of the isotope in a fossil is gradually going to decrease over time. And the less that's left, we know the older it is. And so because we know these break down at a consistent rate, we can determine, well, how much of the isotope is left. That tells us how old that fossil is. Now, we usually don't directly date fossils because to date it we would have to destroy the fossil and grind it up and study the chemical composition of the rock, and we certainly don't want to do that with something valuable like a fossil. In addition, radiometric dating tells us the age of the rock. It doesn't tell us when the rock was formed in terms of sedimentary layers like we typically see fossils. So when we date fossils, we usually want to date volcanic rock because we know that that volcanic rock was formed at a very specific time when the volcano erupted. And so, for example, if we have a layer of rock with fossils in it, if we can date the rock above the fossil, and if we can date the volcanic rock below the fossil, that would give us a time range where we know this organism definitely lived in this particular period of time. And so we're going to use different isotopes to age those different layers depending on how old the layer is. We're going to pick different isotopes. So if we take a quick look at an example, let's say we find a fossil just underneath a layer of volcanic rock, and so we know this rock is something that we can try to date, and the fossil then must be older than that particular volcanic rock. And when we measure this volcanic rock, we see that it has 1/4 or 25% of the normal amount of this potassium-40 isotope. So then we can certainly ask the question, well, how old must this fossil be? Well, we know that our P40 is gradually breaking down, and we said it has a half-life of 1.3 billion years. So that if the fossil was 1.3 billion years old, I'd expect half the amount of potassium-40. If it's 2.6, a fourth the amount. If it's 3.9 billion years old, an eighth of the amount. And so in this case, since I have a quarter of the amount of the isotope left, I know that's 2 half-lives have passed, because half times half is 1/4. And so that means that my fossil must be at least 2.6 billion years old, since it's underneath that rock. And so we can get very accurate dates with these different fossils based on this radiometric dating. It gives us a very clear picture of when events have happened in the Earth's history. Another important concept that really has impacted the history of life is continental drift. And we know that the Earth's surface is made up of tectonic plates, and our continents are on these tectonic plates, and these plates are consistently moving around due to geologic activity, that the Earth's crust is constantly being built and constantly being destroyed. And so as new Earth is built, it pushes plates. As plates run into each other, some of those plates get pushed up, some get down. This is where we're going to see mountain ranges forming. And some of those plates, as they get pushed down, get pushed deep enough, they melt, and that rock turns into lava, and it's going to then be recycled back into another plate at some time in the Earth's history. And so we know all the plates, as they move around relatively slowly for our human lifespan, but over the period of geologic time, this movement makes a significant amount. And we know that historically the Earth's continents have been all joined into one major supercontinent or landmass at 3 different periods in the Earth's history. And so if we look at our different plates here, we're on the North American plate, and the North American plate is moving to the west a couple inches per year. The Pacific plate is moving to the east about 4.5 inches per year, so it's moving a little bit faster. Where these two things collide, then we have the San Andreas Fault. Again, that's where one layer is being pushed up, the other is being pushed down, and every time these two layers push against each other, they store up a lot of energy, and so when those plates move, that's what creates an earthquake. And so we can see we've got lots of different plates gradually moving over our continent's surface. Now, the last time that all of the continents were pushed together was about 250 million years ago, and we can see that at the bottom of this figure here, and that was referred to as Pangea, when we had all the continents kind of pushed together at this last time. And that explains the distribution of many species. For example, we talked about those large flightless birds last time. They arose somewhere about 135 million years ago, when all those southern continents were still pushed together, forming Gondwana. Laurasia was the northern continents of North America and Asia. That was the first split, and then gradually we've seen the continents moving until they've assumed their present positions. And so life has been evolving on top of these continents as they've been gradually moving around. And certainly the position of the continents at different time frames in evolutionary history explains the distribution of species and where we find different species, not only today, but in the fossil record as well. If we take a look at just some of the major events in the history of life, and I don't want to make it seem like we're progressing toward a goal or we're kind of working toward some specific goal in sight. These are just the major steps in terms of the history of life and when they've occurred from oldest to youngest. And your book has a nice example here we see in this figure where it compares the history of life in terms of the time frame of life compared to a 12-hour clock. And the idea is at noon, at the beginning of our time clock, we see sort of the beginning of time and the formation of the solar system. At about 4 billion years ago, we see the Earth formed, and then we can see several different aspects in terms of the history of life applied to that 12-hour clock, going from noon to midnight, for example. And so we know that our first fossils are about 3.5 billion years old. And if we look at this 12-hour analogy to kind of make our time frame meaningful, we see the prokaryotic bar, excuse me, the kind of orange-yellow bar there, starts at about 3 o'clock on our time frame. So for about a quarter of the history of our solar system, there was no life. But about 3 o'clock on our time frame, then we see our first prokaryotic cells. And again, these oldest rocks, by the way, are in Central Australia. The rocks themselves contain lots of these prokaryotic fossils. So the origin of life in prokaryotes is certainly the first major event that we see. And we know these prokaryotic cells are small and simple compared to our cells. And for over 1 billion years, for a large chunk of the history of life, these were the only types of life forms, these single-celled prokaryotic organisms. And in fact, over half of our history of life contain nothing but prokaryotic cells. We know that these prokaryotic cells probably were originally heterotrophic, eating those macromolecules, again, in that sort of primordial soup or that macromolecule-rich oceans. But eventually we see the evolution of photosynthesis, and photosynthesis is the only chemical process on our planet that produces oxygen or O2 gas. And so we start to see oxygen gas appear in the atmosphere and gradually build. And so that tells us photosynthesis was occurring. And again, once we have oxygen in our atmosphere, this is going to change our atmosphere forever, because now we're no longer going to see chemicals being spontaneously formed and being stable in the atmosphere, because oxygen is such a detrimental chemical. And so oxygen being produced by life is the first time we see life really transforming or modifying our planet, which in turn then modifies life after that. And so we see oxygen and life kind of transforming our planet. Now eventually in the fossil record we do see the presence of eukaryotes, and we know eukaryotes are much larger. Our cells have a nucleus, and eukaryotic fossils date around 2 to 2.2 billion years ago. And we know that they were formed by endosymbiosis, that some prokaryotic cells ate others, and those cells that were eaten became the organelles. We talked about that way back before midterm when we talked about our cell organelles. And after our eukaryotic cells arise, we see a huge increase in cell diversity. So looking at our clock here, we don't see our single-celled eukaryotic cells appear until about 7 or 8 o'clock. So again, well after half of our history is gone before we even see our first single-celled eukaryotic organisms. Our next major step then is we do eventually see multicellular organisms appear in the fossil record. Now all multicellular life are made up of eukaryotic cells. They date around 1.2 to 1.5 billion years ago. We can see on our little chart here our multicellular eukaryotes appear around 8 o'clock or 9 o'clock. Our first eukaryotes seem to be some primitive types of algae, so probably photosynthetic. They were multicellular. And after we see eukaryotic cells originate, about halfway through our history of eukaryotic cells, we see a huge diversity, our first really big at what we would call adaptive radiation, where many species kind of appear in a relatively short period of time. And that happened around 600 million years ago or so, and that's referred to sometimes as a Cambrian explosion. And the Cambrian explosion, anywhere from 500 to 600 million years ago, what we see is a huge diversity of organisms appear. We start to see lots of animals appear, different animal groups in terms of marine organisms, and again, for the first time, we start to see life start to take the shape of some of the multicellular life that we see today. Eventually, we do see life on land that's multicellular life. Now, certainly single-celled organisms might have colonized land much, much earlier, but our first evidence of multicellular life on land are plants and fungi moving to land about 500 million years ago after that Cambrian explosion. Animals then followed with things like amphibians as the first vertebrates to really colonize land, followed by reptiles and then birds and mammals as well. And we know modern humans, our own species, Homo sapiens, didn't appear until about 200,000 years ago. So in terms of our little 12-hour clock, that would be with about under a second left of our 12 hours until human appears. And so certainly modern humans have been here just a blink of the eye in terms of the overall picture of the history of life. Certainly one of the main goals then as we kind of walk through these major stages, and certainly we could spend a whole semester on just one of these stages, more or less kind of looking at this history of life, so we're very much taking an overview here. But as we trace history, the field of systematics in biology is the field that tries to classify organisms, tries to figure out what are the evolutionary relationships of species today. How are different species related? When did they arise? How did this history of life kind of unfold, if you will? And today we use the fossil record in systematics, certainly DNA technology. Just like we can do a who's the daddy paternity test to see how people are related, we can look at the DNA of different species and see how species are related. Or historically, we've looked at physical traits, just like maybe a family resemblance in terms of that analogy of looking at your family. Another important branch of biology dealing with this idea of species and the history of life is taxonomy. And the taxonomy is a field that deals with naming and describing different species. We use binomial nomenclature. We use two names when we describe a species, its genus name and its species name. So let's take a closer look, especially at systematics, and this idea of how do we trace or try to figure out the evolutionary history when we start looking at specific groups of living organisms. And if you're in the field of systematics, what you're looking at or trying to generate is what's called a phylogeny. And a phylogeny represents the evolutionary history of a specific species or of a specific group. So maybe I want to look at the phylogeny of humans, for example, and look at our evolutionary history and all of our ancestral species that might have existed. Now, to study phylogeny, the most common form of looking at relationship is called cladistics. And so cladistics is just a way to classify or sort out or to group organisms and try to figure out their relationships with one another. And cladistics is a name that's based on the root word clade. And a clade just refers to a group of species that exist that includes not only the species in our group, but it also includes their ancestral species. So it's one ancestral species, and kind of all of its descendant species would represent a clade. And what we're really interested in when we do this cladistics is to identify what are called monophyletic clades. And I know some of this vocabulary is getting a little heavy duty here. Mono means one, phyletic has to do with phylogeny or one group of species. And so a monophyletic clade would be a group of species plus their common ancestor. So that means all these species are very, very closely related to each other, and they can all trace their ancestry back to this one common ancestor. So in systematics, when they generate these phylogenies, what they're trying to do is construct what are called phylogenetic trees, which is just a diagram that's going to show us relationships and hopefully show us clades, groups of species that all have a common ancestor. Now, phylogenetic trees are just hypotheses. They're like every other branch of science. It's a hypothesis, then we're going to go out and we're going to look at DNA and the fossil record, and we're going to look for evidence to either support or reject our hypothesis. The difference here, though, is a phylogenetic tree is a picture. And so typically, if I'm studying a group of species, I want to pick an outgroup, an organism or species that is a pretty distant relative. And what that distant relative is going to give me some of the ancestral characteristics that might have existed in life before my species ever existed or appear in the fossil record. And then what I want to do is study what are called shared derived traits. And shared derived traits are traits that appear for the first time in the fossil record in a particular group. And if several species all share one of these derived traits, one of these specialized traits, that tells me that those three species are probably really closely related. And they can probably trace their ancestry back to a common ancestor that had that shared trait. And that trait then was passed on from ancestor to descendant species. And let's look at a couple of examples, because I know this gets kind of complex pretty quickly. This is looking at mammals. And within mammals, we know we've got three major groups. We've got the monotremes, which are the egg-laying animals, and the only species really left there is a duck-billed platypus. We've got the marsupial or pouched mammals like the kangaroo, and we've got the placental mammals like ourselves, or in this case it's showing a beaver. So if I want to look at the phylogeny of mammals, I'm going to pick an outgroup or a species that's a pretty distant relative that's not a mammal. So in this case they picked a reptile like an iguana. And so some ancestral characteristics might be things like having a backbone. Because while mammals have a backbone, that's not unique to mammals. Many organisms have a backbone, like reptiles and amphibians. Okay? Having four legs, that would be an ancestral trait, for example. So that's what my outgroup is going to tell me, is what are some of the things that mammals share with other groups that really aren't necessarily mammal-specific. So once I have my iguana, then I want to look at mammal-specific things. And one of the first things we see with mammals that makes us different than other groups is mammals produce protein-based hair, and we have mammary or milk-producing glands. And so it's thought that that common ancestor to all mammals developed those characteristics, the hair and the mammary glands. And so we can see where that pops up on our phylogenetic tree. So that would be a shared derived trait that unites all the mammals. Now, we know that the placental and the marsupial mammals have a gestation, meaning the offspring are going to develop in the embryonic state inside the adult as opposed to egg laying. And so the next branch then would be all the egg layers. We're going to separate out that duck-billed platypus. And so the development of an internal gestation would be a shared derived trait between kangaroos and beavers, for example, marsupials and placental mammals. And then we know that marsupial mammals have a very short gestation period, and most development is done externally in the pouch. So that would be a trait that marsupials share, versus a long gestation where development is completed internally. That would be a derived character for placental mammals. And so we would say that all mammals are a monophyletic group because we can trace our ancestry back to that original mammal that produced hair and mammary glands. Likewise, if we look at smaller groups, all marsupials are a monophyletic group because they can trace their ancestry back to a mammal that first produced a pouch and had a very short gestation period. Here's just another example of one of these phylogenetic trees. Now this is looking at the diversity of all animal life, so this is certainly a very big picture kind of issue. And so, for example, our outgroup might be sponges. Sponges don't have any true tissues, they're just collections of all of the same cell. So the first derived trait we see that's shared between all the other animal groups are having different layers or types of tissues. Then another trait that we're going to see has to do with symmetry. Some organisms have radial symmetry, things like cnidarians, for example, the jellyfish, while other organisms have more bilateral symmetry, meaning if you cut them in half, you have a right and a left half. For example, we as vertebrates certainly have a bilateral symmetry. And so I would say all these organisms, from the echinoderms on down to the nematodes, would be one monophyletic group that can trace their ancestry back to a primitive animal species that first showed bilateral symmetry when that first appeared in the fossil record, for example. To sum up then, we know that there are many different hypotheses that concern the origin of life, but that the hypothesis with the most experimental evidence, the most data in terms of science, has to do with life evolving from non-life, and that these macromolecules developed in these chemical conditions in primitive Earth. We see protobionts forming, which are these phospholipid membranes containing some macromolecules, and then we see the self-replicating molecules, in particular RNA, develop for the first time, and that would be our first primitive form of life. Once we have life, we know an awful lot about the history. We certainly don't know the complete story, but we know an awful lot based on that fossil record. We can date our fossils using radiometric or isotope dating. We know continental drift has played a major role in terms of when and where species have evolved. We talked about some of the major stages in the history of life in terms of the development of prokaryotic versus eukaryotic cells, multicellular life, and so on. And then finally, we looked at how do we trace different species and their history by looking at systematics and generating these phylogenetic trees by determining relationships of different species.